Oh boy, we're almost at double-digit takes on this. Maybe I should make a blooper reel of these. It's not going to be that good. But next couple lectures are all going to be about diversity of macromolecules, the different types of major molecules in our body that perform most of our functions. So, first thing, carbon is the backbone of all of these. That's part of why it is so important for biology, is that it is the main component of the largest and most important molecules in our body. We break them into 4 classes: carbohydrates, sugars, lipids, which are fats, nucleic acids like DNA and RNA, and lastly, protein. Everything needs them. Everything that is alive has them. Luckily for us, most of them are these things called polymers, which are just these huge long chains of repeating subunits called monomers, and it functions basically exactly like a length of chain in real life. You get a bunch of little chain links, you link them together, and with that you can get various different functions that the individual links don't have. Ultimately, we can take a few different monomers and make a huge variety of polymers. DNA and RNA only have 4 each kinds of monomers. That adenine, cytosine, guanine, and, oh gosh, it's not threonine, thymine, duh. Those 4 nucleotide bases make all of our genetic code and get all that diversity. 20 amino acids make thousands and thousands of different proteins. So we can get a huge amount of diversity with only just a few good starting blocks, and ultimately we can predict the function of a macromolecule by analyzing its structure. So let's jump in with carbohydrates. So carbohydrates are a macromolecule. The purpose is for short-term energy storage, as well as providing some building material for cells and organisms, especially plants. That is kind of how they make more of themselves, as with carbohydrates. So the word carbohydrate, carbo meaning carbon, hydrate meaning with water, so carbon with water. Though only elements involved in carbohydrates are carbon, hydrogen, and oxygen. Nothing else is allowed. Only those 3, and they will also always exist in a 1-to-1 ratio of carbon, hydrogen, oxygen, meaning there's always the same amount of carbon and oxygen. There's always twice as much hydrogen as either of those 2. We can see some common ones here. Notice they're in their ring form. They can be kind of a 5-person ring, 5-member ring, or 6. They can also be open form, so in a chain, a length, instead of actually turned into a ring. If we connect 2 of them together, we get disaccharides. So disaccharides are formed by dehydration synthesis, we talked about in the last lecture, which is just a covalent bond that forms essentially and produces a water molecule. They give a special name to it. It's called a glycosidic linkage. Glyco meaning relating to glucose, or relating to sugar basically. Some common examples: sucrose, table sugar, the sugar you're familiar with, is actually glucose and fructose together. And lactose, the thing in milk, is a combination of galactose and glucose. We can make them and break them down kind of at will. However, if we struggle to break them down, we are considered lactose intolerant, which is essentially the inability to break lactose into glucose and galactose for various reasons. We're going to talk more about that later in the year, so I'm not going to stay on it too long right now. If we keep stringing monosaccharides together, we get a polysaccharide, so just many saccharides, many sugars. The word saccharide derives from saccharin, which means like very sweet, something sweet. There are 2 main types of these: storage and structural. We don't really use monosaccharides and disaccharides for structure or storage, because they are kind of just energy units that we need to kind of break down to be stored. So storage polysaccharides, plants make starch, think like potatoes, and that is just a bunch of glucose strung together. Animals use glycogen, which is also a bunch of glucose strung together. We will talk about what the differences in those are, because we cannot actually, well, no, that's cellulose. I will get to that in a bit. But glycogen is actually what insulin tells our body to turn glucose into, so diabetics struggle to convert glucose to glycogen. In order to actually use this energy in these polysaccharides, though, we have to break off the individual subunits. We can't work with the huge long chains. That's why you've probably heard of them as complex carbohydrates, is how they're referred to in nutrition land, because they take time to break down. You don't get that sugar rush and the sugar crash. It's a more sustained form of sugar energy. They can also, again, I say they, organisms, can build structural polysaccharides. Humans don't really do this, but there are some examples in nature. Plants make cellulose, which is, guess what? A bunch of glucose monomers strung together. So just like starch, but cellulose is basically the cell walls. It's what gives plants their rigidity, the snap when you bite into a piece of celery or something. So it is all over. Humans cannot digest cellulose. It is a big component in fiber. Chitin, not chitin, it is chitin, is a structural polysaccharide that sees a lot of use in insects and crustaceans to make their exoskeletons, actually. It is also, I believe, a bunch of glucose, but I think they have some other slight modifications. You can see this stuff sticking up here to make it more useful, I think. I don't really know. Point being, chitin is the animal structural polysaccharide. It's their shells. Ultimately, with all of this, I mentioned and referenced before how both starch and cellulose are just a bunch of glucose strung together. But starch is digestible by us, and cellulose is not. So why is that? It comes down to the bonds in between the monomers, the glycosidic linkages. And also because glucose can take on two different ring structures that I'll show you on the next slide. So the gist is that in starch, all of the glucose is in one configuration, and in cellulose, they're all in the other configuration. We can digest them when they are in the alpha configuration. We cannot digest them in the beta configuration. So this is what it kind of looks like. In the middle here, this is what a glucose molecule looks like if it is unringed, broken out. Then we can see the alpha and beta forms of glucose. So the only difference is in alpha glucose, the OHs here are on the same side. In beta glucose, they are on opposite sides. So when we link them together, it creates this different sort of pattern, where in starch, we get all of these nice little OH groups on the bottom, whereas in cellulose, it's actually alternating. And that alternating is what stops us from being able to break it down, because it gives it a different shape, and our enzymes cannot actually break glucose molecules off of it. So, as I just said, they get a very distinct 3D shape. Starch ends up largely helical, a little bit of branching, not too much, but cellulose is straight and has no branching. The point of this, and also what lends it to be very hard for us to digest, and we can't do it at all, is that it basically aligns the molecules so they can hydrogen bond with adjacent chains. So cellulose will have a bunch of other cellulose molecules next to it and around it, and they'll actually hydrogen bond together and stick together, gives them some extra strength and rigidity, but it also makes them harder to break down. It's kind of like if you want to make a stronger cable, you can do so just by bundling a bunch of that small cable together. Okay, that is carbohydrates. We will go over these questions in class. I'm checking my time. I'm at 9 minutes. We're gonna do lipids as well. And I'm gonna take a quick pause so I can make sure I know what I'm getting into. Okay, talking about lipids. Let's jump right in. So lipids, they're kind of the weirdest macromolecule group because they're the ones that are not made of subunits. The main feature of them is that they are hydrophobic. Like, that is basically the defining feature of a lipid, is that it is a hydrophobic macromolecule. So they are mostly, not all, but mostly just hydrogen and carbon strung together. That gives it very, very strong nonpolar regions. There are a little bit of oxygen and even sometimes phosphorus involved that give them a little bit of a polar section, but they are not polar by default. The ones that do have little polar sections are the exceptions to the rule. And again, they are the ones that don't make polymers. They don't make true polymers, at least. There's not monomers that just bind to each other to make a big chain. We're going to talk about three main types: fats, phospholipids, and then steroids. We're going to get jacked. So, fats, first of all. We're familiar with what they are in food. It is just a bunch of large nonpolar molecules that are called triglycerides, because there are three of these fatty acid chains that are bound to a glycerol molecule. So it's just three fatty acid chains bound to a glycerol, and the purpose is for energy storage. There's a lot of energy in these carbon-hydrogen bonds. These things can be very long, and we can just basically shove a bunch of the energy that we get from other places into this, store it long term. Mammals also will use it to help keep their body temperature up. But that's really all you need to know. Fatty acids made of three chains store energy. They are still made, though, with dehydration and hydrolysis reactions. It's just that you only basically do three of them. One for each of your fatty acid chains. And note here that the fatty acid chains do not need to be identical. They can be different chains, and they very often are, so they can have some different properties. If you see these double bonds, for example, it causes a bit of a different shape, which leads to two distinct kinds of fats. You've probably seen, if you look at the nutrition facts labels, saturated and unsaturated fats, and not ever really known what that means. Well, now you're going to know. A saturated fat means it has fatty acid chains where all of the bonds between the carbons are single bonds. It's all single bonds, no double bonds at all. This is usually leading to them being a solid at room temperature, because they can pack in more densely. And this will be things like butter. Your unsaturated fats have these double bonds. They have at least one double, even sometimes a triple bond between the carbon atoms. These are often oils, so they're usually liquids at room temperature. And the reason they are liquids is because the side chains tend to bend and form these little kinks out that make it so you can't pack them in close together. And so they really don't like being packed together close enough to become a solid. And so they are mostly liquid. The way I remember this is a saturated fat is saturated with hydrogen. It has more hydrogen than an unsaturated fat. An unsaturated fat will have missing hydrogen due to the double and triple bonds. Okay, our next type of fat, next type of lipid, is a phospholipid. You are hopefully familiar with these a bit already. They are the main components of our cell membranes. They are a glycerol molecule, just like all the previous fatty acids, but they have two main differences. They only have two of the fatty acid chains, and instead of the third chain, they actually have a phosphate group. This leads to, combined with the glycerol, a hydrophilic head, so a polar head region, and a nonpolar hydrophobic tail region with the two fatty acids. That is hugely important. You get these special lipids that have basically a polar end and a nonpolar end. This means that when you put them into water, which we mostly are, and our cells mostly are, they will self-orient into a cell membrane structure where the hydrophilic heads will face on the outer portions, so facing the inside and outside of the cell, and then the hydrophobic tails face inward with each other. And this gives us a great barrier that we're going to talk next unit all about. It is called a lipid bilayer, and again, it is the polar phosphate heads orienting towards the outside, the fatty acid tails going inside, hiding from the water. Last molecule, and I think last slide, yes, are steroids. We're going to talk more about steroids when we get to cell communication, which I think is Unit 3. For now, all you really need to know is they're lipids, and you are looking for lipids with these 4 rings that are all attached to each other. They're nonpolar. These rings ensure that. But they're anything from cholesterol to estrogen. So there is a wide variety. We're not just talking about like the steroids you think of people going to the gym and abusing steroids. It's just the class of molecule with these 4 carbon rings. They're very important in cell membranes. We are going to talk about them in the future. Same with the sex hormones as well in Unit 3, so stay tuned for that. But these are nice because they can just go in and out of the cell easily. And that is it for this lecture. I apologize for doing 2 of them. Definitely not doing nucleic acids. It's long enough as is. I'll see you next time for nucleic acids. Woo!